It's my pleasure to introduce this evening's speaker. Sandy Hetherington is an award-winning evolutionary paleobiologist, specializing in the origin and diversification of early land plants. He was a recipient of the Linnean Society's Irene Manton Prize for his doctoral research, and he's now based here at the University of Edinburgh at the Institute for Molecular Plant Sciences. So this evening, he's going to take us back into the steamy, murky depths of the Paleozoic to unravel the origin of spirals in land plants. So you can get your pith helmets ready, and please welcome Sandy Hetherington.
Perfect. Great, just checking that the microphone's all working. Wonderful. Great. Thank you very much for the opportunity to be here tonight. It's going to be a real pleasure to take you on a bit of a journey into a passion for me, botanical spirals. But I want to start off first by saying thank you very much to the Linnean Society and especially Jamie for making this happen. So let's dive straight into it. So plants have an amazing diversity of form. But amongst that form, one thing which a repetitive theme is the spiral. So we see spirals in the leaves of this monkey puzzle tree, in the spirals of the unfurling fronds of ferns, and even if we zoom in on a daisy head, we can see in the middle of the seed head there, our eyes are drawn naturally to spirals. And the aim for this evening is to tell you two stories about the evolution of spirals more generally. So first, I'm going to be telling you a little bit more about the origin and evolution of Fibonacci spirals. And then after that, I'm going to tell you a story about when ferns unfurled. But starting first with the origin of Fibonacci spirals. Before we can jump straight into that, we need to do a little bit of background on a botanical term termed phyllotaxy. So phyllotaxy is the regular arrangement of lateral organs on a plant stem. So we look at the large diversity of plant life alive today, we can categorize lateral organs such as leaves, seeds, flowers into their main distinctive patterns of how they're arranged. We often find them arranged in either an opposite pattern, where the leaves are on opposite sides of the stem, in whorls, where you can see, for example, you can see a layer of leaves and then a separation where you get no leaves and then more leaves again, these nodes and internodes. But one of the most common you'll actually find are where the leaves are arranged in a regular helix around the circumference of the stem. Now, if we actually quantify these different types of patterns amongst plants today, what we find is that spirals are definitely the most common. They occur in over 80% of species. And I highlighted a couple on that first slide, but I'm sure you're familiar with many of the different types of botanical spirals we're used to seeing. We see them in plants like sedums, we see them in aloe vera, in the seed head of a sunflower, or in an artichoke. And many of you will already know that these different spirals really have something in common. And what they have in common comes, we're able to recognize it when we actually start to quantify these different phyllotaxic types. And to do this, I'm going to use the example of a pinecone. So if we take a pinecone, I'm sure if you're looking at it now, your eyes are naturally drawn to some of the spirals you can see in the bracts. So the way we actually go about quantifying it is we take one bract in particular, for example, this starting point here, and from there we can trace either clockwise or anti-clockwise spirals. So in this case, let's start off doing the clockwise spirals. So if we follow from that one bract there and watch it move outwards, we can see the gradual progression of a clockwise spiral connecting together multiple bracts. Now, to quantify it more generally, we can move away from just quantifying that one spiral, but to count up how many clockwise spirals there are in total. This was obviously the first one. But if we look closely, we can see these progressing around the outside, giving rise to 8 clockwise spirals. So the first thing we can recognize is that this pinecone has 8 clockwise spirals. However, we can also quantify the anti-clockwise spirals. So in this case, let's go back to that exact same starting point, but this time, rather than go clockwise, let's go anti-clockwise, shown in the blue colors here, and we can get 1. And then we can progress that further from 1 to then watch these spirals in the blue colors around the outside. And you can see there's many more than there were clockwise, and in total there are 13 of them. So because of this, we term this an 813 spiral phyllotaxy. And what's fascinating, and has fascinated people for a long time, is both the numbers 8 and 13 are consecutive integers, consecutive numbers in the famous mathematical series, the Fibonacci series. And so what we can conclude from this is that we can describe this pinecone as possessing a Fibonacci spiral phyllotaxy. So what that really means specifically is that the number of these clockwise and anti-clockwise spirals, which botanically we will term contact parastickies, are both numbers in the Fibonacci series. Okay, we're going to come back to this in a minute, but we're going to jump out for a very, very brief maths lesson. So just to remind you all about what the Fibonacci sequence is. Fibonacci sequence is a famous mathematical sequence of numbers. You can see it up here, presented at the top, 1, 2, 3, 5, and 8. And in each case, the number is the sum of the two which preceded it. So 2 plus 3 is 5, 5 plus 3 is 8, and so on and so on. It gets its kind of more famous name from the Italian mathematician Leonardo of Pisa, who is also called Fibonacci. But it's been known in maths for much longer than his work. His name just became famous and associated with the sequence. So numbers in the Fibonacci sequence pop up a lot in maths. They pop up a lot, and they're also associated with a number of other factors. And one of those is how they link to what's known in geometry as the golden spiral. So we take the numbers in the Fibonacci sequence, and then we visualize them not as numbers, but as, in this case, rectangles. We can begin to see a pattern emerging, which is shown here. So if we start from the beginning, we can start with a square, which is a 1 by 1 square, and then by a 2 by 2, a 3 by 3, a 5 by 5. And if you do this gradually, building up these squares into this larger rectangle, we begin to get this pattern forming. And if you draw a spiral, which connects together these points, which is shown here in blue, winding its way around, you can very easily make what is almost identical to the true golden spiral of geometry. So this is something you can try yourself on some grid paper if you've got some. A go at actually being able to draw yourself a golden spiral. And these golden spirals pop up in all kinds of things. They're famous in art and architecture. And so it's quite amazing that it's the Fibonacci sequence that links to it. Okay, so just to summarize again, the Fibonacci sequence is this famous mathematical sequence. It pops up and is related to a variety of things, including the golden spiral. And what's particularly interesting for us is that it seems to be present in plants, and it's present in plants frequently. So I gave you the one example of a pine cone, and I find that example quite useful for identifying and describing what the Fibonacci sequence looks like in plants. But you shouldn't be convinced by just one single replica of anything. This could just be the anomaly. So what we really need to do is to not look at a single pine cone, but to extend this analysis out to a much, much wider investigation. Maybe 200 pine cones. Now, I'm not going to sit with you and gradually go through each of these. And we're very lucky to know that actually someone has done this for us. So we can skip from 200 pine cones to 6,000 pine cones. In a study by Ferns from 2015, they analyzed the phylotactic patterns amongst 6,000 pine cones of this pine tree here. And by doing that, of a single pine tree, they were able to identify that over 5,838 of them, so that's 97% of all of those pine cones they examined, were representing as 813 spiral phyllotaxy. So these things are common. Well, they're common in pine cones. However, as we continue our search wider afield, we find that they're common in more things. So each of those examples I showed you earlier, whether it was the succulent, the aloe vera, the sunflower, or the artichoke, they are all types of Fibonacci spirals. The aloe vera had the 8-5 pattern, the sunflower 21-34, and the artichoke 34-55. So they're common, and actually, if we go about taking a more systematic approach from this, which is exactly what Jean did in 1992, compiling together the investigation of almost 13,000 observations, which is accumulated data from over 650 species, he was able to identify that over 91% of these species he examined had Fibonacci spirals. So they're incredibly widespread. But one of the things that I find most fascinating in them is they're not just widespread, but it seems to be almost any trait associated with phyllotaxy that you investigate. Fibonacci numbers in the Fibonacci sequence just seem to pop out. So up to this point, I've been telling you about what happens when we take a centric approach, which is, as I said from here, it's the kind of most simple. If you pick up a pine cone, you can do it yourself. You can look down at the spirals radiating out from the center. However, a separate approach you can do is called the cylindrical approach. So rather than, which is shown here in the middle, rather than seeing these spirals radiating out from the center, we can actually try and describe the arrangement of leaves on an overall cylindrical stem. So the way we typically go about doing that, which is shown here in this nice idealized reconstruction, is to recognize two leaves or two flowers or any of these two lateral structures that sit directly above or below each other. So in this case, we can take this point at the beginning up here and this point at the bottom here. And what we then want to do is to count up the number of leaves that separates them and the number of times you have to go round the stem to connect them. So in this case, there are actually 8 leaves that separate them. And to connect these points, we have to go 3 full times round the stem. So this is a 3-8 helical phyllotaxy. And guess what? 3 and 8 are sitting there in the Fibonacci sequence. And obviously, this is an idealized model. But again, if you go out and investigate a huge variety of plants in nature, you get these same patterns popping up again and again. And for me, I think the most fascinating of all really comes down to investigating the divergence angles of newly developing primordia. So when we investigate plants, they grow from apical structures to meristems. These are self-renewing structures which allow them to grow and produce new structures like leaves and flowers. And this image on the right-hand side, what we're looking at is a zoomed-in microscope image of the growing structure, the fertile inflorescent structure of Arabidopsis thaliana. This is a model species that we work on a lot in plant sciences. And what is drawn onto this, if we have the shoot meristem where the population of self-renewing cells is at the center, and it's producing these new primordia, and we've numbered them in their order. So number 1 was the most recent, and then before that was number 2, and we can gradually move around and move further outside. But what's interesting is actually the Fibonacci sequence itself gives us a prediction about where new primordia will form and the angle that connects existing primordia. And we can illustrate this by having a look. I've drawn on with this black line, the angle connecting primordia 1 and primordia 2. And so what we're going to do now is a tiny bit of audience participation. So if anyone's got a phone with a calculator on, could someone please get one out? And what I'd like you to do is pick any of these numbers up at the top here, any of the numbers in the Fibonacci sequence, take 2 consecutive numbers. So for example, we could take the number 34 and 55. Could you please take 34 and divide it by 55? And if someone could give me, maybe shout out what sort of numbers they're getting from doing something like that. Yep, and then because we're interested in the overall angle, let's take that and then times it by 360. So we can actually get an angle. So what that fraction is represented in terms of an angle of a stem. And so what we're able to do, getting that number is that right? Everyone getting numbers close to 222.5? So what you've just done there is actually calculated, and you can go in and measure this on a huge variety of plants today. You'll find that the angles that separate 2 newly developing primordia will be those values. So the large value will be 222.5, which leaves you the small value of 137.5, the famous golden angle in geometry. So amazingly, it's this Fibonacci sequence. It just seems to keep popping up in so many different aspects of phyllotaxy. And this has really fascinated people for a very, very long time. And in particular, I think many of you might be sitting there and thinking it as well. The question that really springs to mind is, so why on earth are Fibonacci spirals so common? And if you're thinking that, you are in good company, because it's a question which has been fascinating scientists for years, and even fascinated and bothered Charles Darwin for a while. So after publishing his Origin of Species, in around 1860s, Charles Darwin became fascinated with phyllotaxy. And there were 2 main approaches he was using to investigate phyllotaxy, but the one he was using most was this cylindrical approach I described to you earlier. So if you remember it, as I described it earlier, it's when we want to work out, we want to describe the positions of leaves that are directly above each other. So we've got the 0 and 8 here, and we could describe this as a 3-8 pattern because it's been around 3 times and separated by 8 leaves. Okay, so to investigate this, rather than having to draw out these quite complex 3D diagrams, the way we often visualize the cylindrical patterns is to imagine wrapping a sheet of paper around the outside of the stem and marking on the positions of the leaves. So similar to what's kind of shown in this illustration here. The advantage of doing that is it's then very easy, if you lay that piece of paper flat, to have a 2D representation of a quite complex 3D pattern. So you get the dots represented on here. And this is exactly what Darwin was doing. What he was trying to do was to visualize the distribution of the leaves on a plant stem, and doing that idea of wrapping it round to get the cylindrical approach and then unwrapping it and viewing it flat. And there's two examples here. What's amazing is obviously we can still look into Darwin's notebooks and see his own drawings and his own doodles on these. And so in particular, what we wanted to do was to, in this case, to examine the distribution of leaves, separating this leaf here and this one here. And so by doing that, you could count up the leaves that separate them, as we've said before. So this gave 5 leaves separated them. And to connect to those 2 points, you had to go round the circumference of the stem twice. So this pattern is a 2-5 pattern. And as you've already guessed, these are in the Fibonacci sequence. And yeah, it's a 2-5 phylotactic pattern. And then at the same time, when he was doing this, he was thinking to himself about the different types of phylotactic patterns that could exist. And this is what he's showing on the next slide. He drew an example which looked almost identical to the one on the left, but just slightly different. And if we do that same analysis again, in this case, he found that this example would have a separation of 4 leaves, and you'd have to go around the circumference of the stem 3 times to get to it. Now, this is a bit confusing, because what we're getting here is a 3-4 phylotactic pattern. 3 is in the Fibonacci series, but the number 4 isn't. Okay, so what bugged Darwin about this is that he could go out into the garden and examine a variety of the plants. And when he did that, what he would find is that the examples where the numbers were in the Fibonacci series, for example, the 2-5, as he's written in his note next to it, are real. As in, he can go out and find plants, and there's lots of examples of plants that have that. However, this 3-4 example, the one where it's not in the Fibonacci sequence, as he's written next to it, is imaginary. He's gone out looking for it and therefore, but hasn't been able to find a single example of a plant that has that pattern. So how could you have these 2 patterns that look superficially so similar, and yet one is always found in nature and one is not? And this bothered Darwin a lot. And I think he summed this up best in a letter he wrote on the 11th of May, 1863, to the botanist Asa Gray. I've been drawing all the real angles and unreal angles on a spire, as we've just been talking through with them. And I see that the angles which do not occur in nature are just as symmetrical in position as the real angles. If you wish to save me from a miserable death, do tell me why the angles of 1/2, 1/3, 2/5, 3/8, which I'm sure everyone in the room now knows as the numbers in the Fibonacci sequence, why do these occur and no other angles? It's enough to drive the quietest man mad. So the issue was, at this time, Darwin was developing, or had developed his theory of natural selection, a mechanism for producing an endless forms most beautiful. But why then did what seemed to be an entirely reasonable set of forms, these 3-4 patterns, which he put in his notebook, why did they not exist? And yet always the patterns that were in the Fibonacci series did exist? This was a really big question, and it bothered Darwin that he wasn't able to get a satisfying answer. What was the adaptive significance of this, which was lacking on this side here? So it fascinated Darwin, and it's fascinated people since Darwin, and we still don't have a definitive answer. One of the most widely cited that if you open a plant textbook, one of the first things that you might read is it might say that these Fibonacci spirals are associated with light capture. This is an idea that goes all the way back to Leonardo da Vinci, with the prediction that leaves avoid self-shading by being positioned in this way. However, in recent years, this has been largely debunked. And the reason for that is we know from modeling approaches that there are a number of other angles that would allow plants to avoid self-shading. So that's one line of evidence. And then the most obvious of all, if you talk to many botanists, they'll know that plants have such a plethora of different mechanisms for changing and capturing light. They can angle their whole leaf, they could extend their leaf blade. It's not typically them making major changing with their phylotactic pattern, which is enabling them to catch more light. Okay, so we're not sure about that one. The next major hypothesis being put forward is the theory that maybe it's actually incredibly adaptive for packing. So if you want to put in as many seeds into a seed head, you want to put in as many primordia at the apex, which is shown in this middle section here. Again, there's some evidence, but again, it's not completely compelling and it's not completely ubiquitous across plants. And then the final one, which has been hypothesized as well, is actually maybe there isn't in itself the one clear adaptive solution, but maybe this is simply just a self-assembling phenomenon. So what we mean by that is that if you have a shoot apex and it's developing lots of these smaller primordia, maybe they naturally fall into this Fibonacci sequence. Maybe it's linked with the fact that plants germinate from either seeds or spores, and during their development, as they get bigger and more complex, that they actually are producing these Fibonacci type patterns, or maybe it's something to do with their underlying genetics. The bottom line is we are still unsure. Each of these sounds could be reasonable, but actually narrowing it down to a small number a small number of actual testable hypotheses has been really difficult and has been a really challenging thing for scientists for centuries. And it's something which really fascinates us in my lab at the moment. And so one of our key focuses at the moment is to begin to shed new light on this question. So we recently started a grant called YFib, so why are Fibonacci spirals so common? And the approach we're planning to take is, rather than taking a single one approach, we're planning to take an interdisciplinary approach combining evidence of fossils, living plants, and genetic networks of living species to see whether any of these taken together might give us an answer that taking one in isolation might not hold us. And what I'm going to do now is give you an example of what we've learned from taking a look at the fossil record of plants. So we're going to leave present-day Edinburgh behind. We're going to take a trip back to the Paleozoic. So the Paleozoic is the block of time between 250 to 540 million years ago. It's a key time in plant evolution. It's when plants radiated across the terrestrial surface and transformed the Earth into the green planet we know today. This story of plant evolution starts 470 million years ago, which is our first direct evidence of fossil plant spores. From this point, though, we had to wait, sadly, another 40 million years until we actually find out what kind of plants produce these spores. In this case, they're these tiny branching stems which look completely alien to us today. These plants are often grouped into the genus Cooksonia. They just have these branching stems and these terminal reproductive structures. So it's kind of hard to imagine what a world would look like, but the amazing artist Richard Bisley has tried to capture what a world during the Silurian must have looked like, with vegetation which would barely be scraping your ankles, in plants that look completely different to what we're familiar with today. There's no leaves, there's no deep roots, just these strange twiggy plants. And what's crucial, though, is without any leaves or lateral reproductive structures, there's no spirals, and there's nothing we can characterize by taking a phylotactic approach. We need to wait until the early Devonian. So we cross over the geological period into the early Devonian, roughly 410 million years ago, and this is when we start to see plants which look more familiar as plants today. This is a coaley compression fossil described by Matsunaga and Tomascu of an early lycopsid. And what you can see are these little spiny leafy structures or leaves on the outside of this stem. And it's the first time we can actually find clear evidence of spirals in the fossil record. You can see they've marked on the spirals here. You can see the consecutive connections of these leaves. So we have spirals at this point in time, but the big question remains, are these Fibonacci spirals or are these a different type of spiral pattern? And we're actually very lucky here in Scotland to have the fossil site where it's possible to investigate this in more detail. And that fossil site is known as the Rhynie Chert. So the Rhynie Chert gets its name from a small village of Rhynie in Aberdeenshire, located up here. And if you go there today, you're far more likely to find sheep grazing on this very, very important scientific site than any fossils. So we visited around this time last year at the end of November. And this is us standing in this incredibly important scientific site with not a fossil in sight in the kind of squelchy mud. But it's a kind of place of pilgrimage you have to go if you work on fossils from this point in time, because it's just such a unique site. Because what's actually important, I'm holding an example in my hand in this picture, but about 6 feet below our feet, there are a series of rocks known as the Rhynie Chert. And these rocks, the Rhynie Chert, give evidence to the fact that what is now a kind of damp Aberdeenshire countryside was once an amazing environment with exploding geysers, bubbling hot springs, and a diversity of early plant life, again captured in this lovely reconstruction by Richard Bisley. So the Rhynie Chert gives us insight on what life was like in Rhynie 407 million years ago. And we've been doing a lot of work on the Rhynie Chert because it's just a fascinating place with exceptional levels of preservation. And in particular, the plant that we've been drawn to most in the Rhynie Chert is this one here, Asteroxylon mackii. Compared to those small Cooksonia type plants, this is something which I think if you were walking across the vegetation, you'd know that this is a plant, it looks like plants do today. It has a complex underground rooting system, it has some prostrate stems, and then it has these complex leafy shoots which extend upwards from there. And so for us, this seems like the ideal system to go and investigate in more detail the structure of the leafy shoot. So that's precisely what we did. We turned to the leafy shoot of these plants, and our aim was to characterize phyllotaxy in this plant, giving us the earliest record of phyllotaxy in the fossil record. So this was work started a number of years ago when I first moved to Edinburgh by a brilliant undergraduate student called Holly-Ann Turner. So Holly-Ann joined the lab during COVID and we needed a remote project to work on. Unfortunately, I said, I've got some digital scanned images of some series of fossils known as peels from the Rhynie chert that look something like this. I said, if it's possible, you can do this remotely to actually take these peels, each one of them almost like sellotape, you can imagine, a very, very thin sheet. If you're able to take these digital images, you can actually digitally reassemble them into a 3D model, sticking them back together to produce the fire taxi we could see then. And this is exactly what Holly-Ann did. She started off in her undergraduate and then continued as a research associate afterwards. So what she was able to do was to build for the first time 3D models of exactly what these plants would have looked like. So it was the first time we could actually visualize a plant stem of something that was living 400 million years ago, and we could recognize the arrangement of leaves around it. They're only a few centimeters in size, but still gave us this unique insight into early plant life. The largest specimen she examined was this one here, which is almost 5 centimeters in length, and really gave us a chance to, again, really visualize the arrangement of leaves around the outside of the stem. So this allowed us to take that centric approach where we could investigate the arrangement of leaves around the outside. But combining these, one of the reconstructions she made was actually of an apical portion. And so by taking that 3D model and working really closely with Matt Humpage, one of the authors of the study, who's incredible at making digital 3D renderings, he was able to actually take her model and produce this incredible top-down view of what that apex would have looked like. And we were able to make a digital slice through the apex. So in the same way that we're able to characterize the phyllotaxy in the pinecone, we could do that here. So we combine that with some additional evidence, such as some incredible fossils like this one here at the Hunterian Museum in Glasgow, which you can clearly see evidence of these spirals radiating out of the center in this plant. Okay, so taking this diversity of new fossil evidence, what we were able to do was to take those two approaches I described to you earlier. Take the cylindrical approach, take the centric approach, and let's quantify phyllotaxy. So what we found is the cylindrical approach. So we were able to calculate the arrangement of the leaves on the side of the stem, and then taking the centric top-down approach you can see over here. And what we were able to find was, from this example and combining all the data together, that this one species seemed to have quite a large diversity of different phyllotaxic types. Actually, quite a remarkable diversity, actually. There seemed to be evidence of whorl-type patterns. This is the one I mentioned earlier, where you have a layer of leaves followed by an internode with no leaves, and then another layer. You can see this in the 3D reconstruction. And when we're looking at it in transverse section, we notice it when the two numbers are the same. So the 5-5 and the 6-6 patterns. And then on top of those, we found large numbers of spiral patterns shown over on this side. You can see the blue spirals forming these helices around the outside of the stem. And what's interesting is that all of these spiral types all conform to the same overall pattern. They were what we would call an n to n+1 pattern. So that means 4 to 5, 7 to 8, or 8 to 9. And for the eagle-eyed among you already, what you might have already spotted is that these numbers, therefore, cannot be in the Fibonacci series. And actually, these are actually some of the types of numbers which are Darwin's imaginary type of phyllotaxy, the ones that we just didn't expect to find in nature at all. And here they were in this early fossil. So what we would begin to realize is at the earliest point in time we could characterize phyllotaxy, we were able to see these very rare types of non-Fibonacci spirals, these n to n+1 spirals, different from the vast majority of living species today. Now, alone, this was really interesting. It gave us a chance to have identified how a fossil could begin to change our perspective on the evolution of phyllotaxy. But actually, I think the most significant part of this was to not just look at the fossil in isolation, but to combine it with living species today. It gave us a new perspective on the living lycophytes. So if we look at a very small family tree of this group called the lycophytes, Asteroxylon, the fossil, sits down here, and it's sister to this living group called the Lycopodiales, which are alive today. So we thought it's definitely time to take a re-examination of the Lycopodiales. And by doing that, what we were able to find is that many species of Lycopodiales, including the fir club moss, which you can find around lots of bits of Scotland today, Persia selago, also have a highly variable phyllotaxy, often display both whorled and spiral phyllotaxy, and also have regular transitions. So all of these characteristics that we're finding in Asteroxylon are found amongst this living group of plants today. But the big difference was actually our own change in perspective. Previously, this group, the Lycopodiales, had always been treated as an oddball, a lineage that had gone out on its own to do something really weird. However, with this fossil data, what we're able to now conclude is no. This is something which seems to be in lycophytes and highly conserved. So it's been highly conserved for over 400 million years amongst this group, which gives us this unique perspective on the fact that not all plants develop Fibonacci spirals. And in fact, in this group, the Lycopodiales, there's clear evidence of conservation from these ancient fossils such as Asteroxylon all the way through to living Herpetia. And I think one of the really nice things we could do in our study is actually take those 3D models and 3D print them. So although these plants will never cohabit together, we can actually show what it would look like to scale having our fossil Asteroxylon next to a living member of Herpetia. Okay, so what Asteroxylon gave us was this new perspective on the evolution of Phyllotaxis in this group. And what I like about this most is actually I think it posed many, many more questions than it answered. Because actually, by finding out that there was this group, Lycopodiales, which had been overlooked developing non-Fibonacci spirals, it's made us think more about, are there other groups of plants that develop non-Fibonacci spirals? Or is there something different going on in lycophytes compared to other euphylophytes? And then finally, there's quite a nice prediction, which is to think, well, we live in a Fibonacci world today. The vast majority of plants are Fibonacci. Was there a period of time in the past where we could be living and actually the plants not show that? In a lycophyte-dominated world, would it have been a non-Fibonacci world? And these are all questions we're hoping to answer in the future. Okay, so to draw to a close the first story about the evolution of Fibonacci spirals. What I hope I've convinced you about today is that Fibonacci spirals really are incredibly widespread in plants today. The reason for the high frequency still remains uncertain and is a really major evolutionary question, and one that we're hoping to contribute to in the future. And then importantly, in our group, we're particularly interested in this unusual group of plants, the lycophytes, because they seem to be a great case study in a group of plants that's existed for over 400 million years, and yet they have a really high prevalence of these non-Fibonacci spirals. Perfect. So with that, I will move on. And for the final part of the talk, I want to tell you a story about the evolution of ferns. So this story started and being carried out by a brilliant postdoc in the lab, Dr. Raphael Cruz. And both Raphael and I are fern enthusiasts. And a big question that we've both been pondering is, when did ferns first start to unfurl? So as many of you will know, fern leaves develop gradually and uncurl from structures known as fiddleheads or croziers. And you can see why, due to their close similarities in the form between these two. But in terms of botanically, we often term this as cernate leaf tips. And they really are a kind of iconic feature of the group. They feature in art. They're often a symbol of kind of new life, such as this sculpture at Doit Botanic Gardens, close to Edinburgh. And I think with that goes hand in hand this idea that it's often ferns that pop up first after there's been either catastrophic fires. They're some of the first things to pop out and unfurl. And not only are they important for this, but in actual fact, they also have given rise to a bit of an icon in the UK, the decoration on the custard creams, which are recognized as being inspired, these custard cream biscuits inspired by unfurling fronds. And I would highly recommend, if you want to know more about that, to follow Manchester Museum's social media and hear a bit more about that. Okay, but for us, we're particularly fascinated in their diversity. They're incredibly diverse. There's not just one single type of fiddlehead. Each plant does it in a slightly different way. From minute ones that are a millimeter or so in size to things the size of your fist. And with this diversity, therefore came the question about when these structures evolved. And this is what Raphael and I have been wanting to get our teeth into. So we know today that ferns comprise about 12,000 living species, and molecular phylogenies mean we have quite a good understanding of the overall structure of the fern groups, knowing that they're separated into 5 major clades. So what we wanted to do was to take a systematic approach, go through group by group, and work out whether they have fiddleheads or not. So we started with the early diverging members of the group, the Equisitales, Ophioglossales, and the Stylotales. Groups that you might not be familiar with. So here are some nice pictures from the brilliant Ferns and Lycophytes of the World project. The Equisitales are more commonly known as the horsetails. Ophioglossales you might know as the adder-tongue ferns, and the Stylotales, they're a very unusual fern, often called the wisp ferns. Okay, these are these 3 very distinct groups of ferns, but what's distinct about all of them is that none of them seem to have any evidence of fiddleheads. And this is true not just at the macro level, but at the micro level too. If we zoom in on the apex, the growing apex of any one of these, we can't find any evidence of fiddleheads. This is, if you look at the shoot apex of the horsetail here, you can see the leaves, they curve over the top, but they never form that type fiddlehead we're familiar with in many other groups of ferns. Okay, so this really surprised me. My impression going into this was that many of the groups of ferns, they'd be producing fiddleheads, and yet there seemed to be quite a convincing argument here that all three of these groups lack fiddleheads, and we found no clear evidence of them, even in their kind of evolutionary history, as developing fiddleheads either. So let's leave behind this group and instead focus on the other group of ferns, the Maritales and the Leptosporangids ferns. So this includes the vast majority of living fern species alive today. And from an examination of these groups, what we're able to recognize is that basically all of them produce fiddleheads. This amazing diversity, if you see a fiddlehead, it's likely to be in one of these groups, and it's most likely to be in the Leptosporangid ferns, as these are the most common today. But what was quite fun is that previously we noted that there hadn't actually been a name given to this group, the grouping of the Maritales with the Leptosporangid ferns. And so Raphael was able to come up with the name the Cercinatophytes, or the fiddlehead ferns, to describe this as an informal grouping of ferns grouped by the characteristic of fiddleheads. So for us, this was really nice. We were able to recognize this as a feature which defines a group, an ancient group of ferns, and then poses this next question, which is, okay, how far can we trace this group back into the past? So again, we're going to take another dive away from modern day, present day Edinburgh, and this time go back not 400, but 300 million years. So roughly 310 million years ago, if we're standing in Edinburgh, it would have been in the middle of a coal swamp forest. This is another one of Richard Bisley's brilliant reconstructions of what that would have looked like. This giant swampy environment with enormous arthropods, and it seemed like an ideal place to go hunting for some fossilized fern fiddleheads. The reason for that is we know that ferns were abundant in these ecosystems, but it was a bit more unclear as to whether fiddleheads were present. So what we did is took a deep dive into the literature, and in particular, we attempted to characterize some of the literature documenting these carboniferous forests and the ferns that were in them. And by doing that, we came across a number of specimens which have been assigned to the genus Spiropteris. Now, paleobotanists do something quite strange, which is when they find a bit of a fossil which looks distinctive, they'll often give it its own specific name, which within the Linnean Society is talking about this is a bit more problematic as to how we can actually recognize and interpret these as two species. But we do it in fossils because we're used to finding these bits of plants and not being able to stick them together. And so we call this a form genus, Spiropteris, described by Kidson as a really nice example. And you can see in his early diagram, it looks almost identical to what we see in modern fiddleheads. But what's really nice is that not only can we go and read Kidson's paper, we can actually go back and actually look now at Kidson's collection itself, because the vast majority of it is in the British Geological Survey. So we can actually go and examine these specimens again, which were the ones that Kidson used to describe these. And what this told us is that at least 300 million years ago in the UK, there were clear evidence of fiddleheads, but we weren't able to assign them distinctly to a distinct group of plants. To do that, we needed some exceptional levels of preservation. And we're very lucky to have this in this one example in the museum collections in the Natural History Museum in Paris. This is a wonderful image, very kindly taken for us by Cedric, showing an example of a brilliantly preserved fiddlehead from just over 300 million years ago. Now, the difference is that in this case, we have exceptional preservation. You can see these multicellular hairs, which allow us to distinguish this and actually associate it with other bits of fossil material from the same site. So we know that this is actually a member of Botryopteris. So we know it's a fern and we know it's got fiddleheads. So taking these 2 points together, we could conclude that fiddleheads have been unfurling for over 300 million years, which is really quite exciting. But a bigger evolutionary question we had was then to ask, okay, but where did these structures come from? And this really touches on a big motivation I mentioned earlier in the talk as well, just to begin to understand when leaves evolved more generally. So to do that, the kind of question we're really asking ourselves is how you go from these tiny alien branching twiggy Cooksonia things into plants that we know with much larger, much more complex leaves. So to put that just on a very, very simple illustration, how do we go from this type of plant to the modern relatives today? Well, the major theory in the literature for predicting how we undergo this change is a series of steps which has been collectively called the telome theory. It allows you to gradually modify these early plants here, these little naked branching stems, to produce a leaf. You've got a couple of key steps involved. The first thing you need to do is you need to start having some of your branches as small branches on the side, which is known as overtopping. Once you have some small side branches, you need those the branches within that side branch to all branch in the same plane, giving you a nice flat surface for capturing light. And then finally, you need to produce some tissue, which is going to fill in those gaps and again, improve your ability to capture light. Sounds like a nice theory. There's some evidence to support it, but it's always been a bit of an issue about how you'd actually go about testing this in any sort of formal or meaningful manner. So one way you might be able to test this is to think about the similarities between shoots and leaves. If leaves evolve directly from shoots, we might expect them to have close similarities with the shoots we find with shoots. And so what we want to do is to therefore make a comparison between the shoots and leaves of living ferns and see how similar they are. So if we zoom in on a shoot of a living plant today, this is that equisetum apex I showed you earlier, but this could be many fern apices, they all look broadly similar. At the very, very center of the apex, there's typically a large triangular cell. It's triangular when it's cut in longitudinal section like this, but if you're able to view it from above or to produce a 3D reconstruction, you find out that triangular cell is actually tetrahedral in shape. And what's amazing amongst the ferns is that these structures are ridiculously highly conserved. And again, they can be found in fossils. This is another amazing example of a 300-million-year-old fossil, which has that really big prominent tetrahedral cell at its apex. So to actually go back to the T-lone theory and actually frame this within a hypothesis, we might predict that if the T-lone theory is correct, we might expect to find these tetrahedral cells in both shoots and in leaves. And that's something we wanted to investigate further. So the first place to start is to look at a diversity of leaves in living species today. Now, one species to potentially focus on could be Osmunda. And the reason why I think Osmunda is a great one to talk about is that if we were giving this talk actually in Burlington House in London, if I was standing here, I'd be looking almost directly at this portrait here of Irene Manton. So she was the first female president of the Linnean Society, a fern enthusiast, and in her portrait, she's actually holding on to Osmunda. So what's particularly nice about this on a talk about botanical spirals is just to very quickly note that she was particularly interested in Osmunda, and she actually recognized that the chromosomes of Osmunda, which is what Irene Manton was most famous for, were actually also spirals. This is one of her micrographs from 1949 showing this. And so it's just a nice excuse to talk about the brilliant work of Irene Manton. But I digress. So we took Osmunda as a great species to look at. If we cut a longitudinal section through the fiddlehead and then look and zoom in right on the very apical point, we find another one of these big triangular cells in longitudinal section. But here's the big difference. When we look at this now and make a 3D model of this, we don't find it to be tetrahedral. Instead, we only find it to have two cutting faces and to be almost like a doorstop shape, a wedge-shaped cell. So what this tells us is from looking at these species that we have tetrahedral cells in the shoots of living species, whereas we have these wedge-shaped cells in their leaves. So it's quite a big difference and therefore doesn't necessarily support T-lone theory. But again, this is only based on comparisons of living species. So let's turn to the fossil record and see what that can tell us. Again, to do this, we really need a site like the Rhynie Chert, a site with exceptional preservation, but this time from the Carboniferous. And we're very lucky in the UK to have one of these sites, which are known as Carboniferous coal balls. You can see how they get their name from their ball shapes. And they were found extensively in the Lancashire and Yorkshire coalfields during the Industrial Revolution when people were mining coal. Now, what's incredible about these coal balls is when you slice them into thin sections and examine them under the microscope, they're preserved with exceptional levels of preservation. And it had been reported just over 100 years ago by Bauer, one of the leaders of work on ferns more generally, in his book on the ferns in 1923, of an example from one of these Carboniferous thin sections of the apical point of a leaf. And again, what's amazing about this is this specimen was in the Kidston collection, and therefore we could actually go and have a look at this specimen again. So this specimen is now in the Hunterian Museum over in Glasgow, and a big thanks to Neil Clark for allowing us to look at it and re-image it. And what you can see is this amazing fossilized fiddlehead, preserved with exceptional preservation, including the cellular preservation of it. So you can see the fiddlehead structure there, and we work with the brilliant botanical artist, Julianne Keeley, to be able to produce a reconstruction of what that fern would look like if we were in the room today. But for us, as I said, we were particularly interested in the apical point of these cells. So we could zoom in on the apical point, and as Bauer had already noticed, at the tip, there seem to be these large triangular cells. And this is where the issue comes. And this was the issue for Bauer previously. As I mentioned, if you cut a longitudinal section and find a triangular cell, it's impossible just with that one plane of section to work out whether it was either tetrahedral or wedge-shaped. And that was the issue and the previous stopping place for all previous investigations. But what's nice today is we can take the same specimen and examine it under the confocal laser scanning microscope. So this is an instrument which underpins most of molecular biology. It's one of the workhorses of molecular biology today. We can use a laser to excite the specimen and get autofluorescence off that original biological material, and therefore not just collect information on a single plane, but to build up a 3D projection, which is exactly what we're doing here. So we could now look at that cell both in longitudinal section, but now also look at it in transverse section as well. So that allowed us to investigate whether there's evidence of it being a wedge-shaped cell or a tetrahedral cell. And in this example here, what we could clearly see is that if it was wedge-shaped, we'd only expect to see this lozenge shape, only two, you get to see the two sides of the cell. Instead, if it's tetrahedral, we'd expect to see this triangular outline, just like we see here, which seem to clearly support the fact that there seemed to be a tetrahedral cell in the leaves of this fossil species. So what was particularly interesting about this for us now is it changed our perspective and actually put light back on the Thielen theory as being a potential mechanism for explaining how leaves evolved. We know we have tetrahedral apical cells and shoots. We now had evidence that in early groups of fossils, early groups of ferns, including this new fossil, there's tetrahedral cell in the leaves as well. And therefore, it must have been a later innovation, this switch to a wedge-shaped cell on those on the osmunda leaves that Irene Manton was holding on to. Okay, so taking this all together, we could actually build a new picture for how leaves evolved in ferns. We can picture an ancestral shoot where there was a tetrahedral apical cell, and from that ancestral shoot, leaves first evolved, and they also had a tetrahedral apical cell. From this point onwards, we then get the origin of fiddleheads. So our new group, the fiddlehead ferns, evolved. And then we get the early diverging branches of those, such as the Maritales and the Osmundales. And around this time, over 300 million years ago, we've actually got evidence of these tetrahedral apical cells in these fossils. And therefore, it was only a later innovation to switch between a tetrahedral and a wedge-shaped cell, which then gave rise to the vast majority of the leptosporangiate ferns we're left with today. And so with that, that just brings me to the end of the talk. So I hope I've taken you on a bit of a deep dive through the evolution of botanical spirals and demonstrated as well how fossils can be really key in changing our perspectives from just looking at living species. So to wrap it all up, what we can say is very, very early in land plant evolution, the common ancestor of vascular plants appeared to be leafless. There was no evidence of spirals, no evidence of spirals at all, and no leaves. And then from this ancestor, the lycophytes and the ferns split. In the case of the lycophytes, they went on to do something quite unique in the production of these non-Fibonacci spirals that we see in the case of Asteroxylon. In contrast, in the ferns, we get the origin of this unique structure, the fiddlehead. And the fiddleheads occur in ferns, and what we were able to demonstrate is that was likely through a modification of a pre-existing shoot with a tetrahedral cell. And so taken together, that begins to unpick how fossils can really, really display about how spirals were evolving and have changed through time. And so with that, I just want to end by saying I'm based in the Institute of Molecular Plant Sciences up at King's Building. We do a whole range of different outreach events, and I really highly recommend if you've enjoyed this talk, want to hear a bit more about the kind of work we do of some of the other groups as well, please do follow us on social media or read some of the blog posts coming out of our institute. And with that, I just need to say a huge thank you to my funders, to my amazing lab group, and in particular to the Linnean Society and Jamie for organising this event. I'm very, very happy to take some questions. Thank you.